What is GAPS?
The genetic analysis of Pacific salmonids. It was a baseline which colleagues from California, Oregon, Washington, Canada, and Alaska collectively created, characterizing fish from each of our regions so that we could ID fish back to their most likely source. We developed this standardized DNA baseline which we offered as a tool to be used alongside the coded wire tag tool for learning about fish in the ocean. There were actually 12 genetic laboratories involved: Canada, Oregon, Washington, Alaska, various states in the US, Idaho also. And we chose 13 different microsatellites as the base data. Those 13 microsatellites have of order of about 500 different alleles, more than 40,000 salmon assayed across that large region so that we could know the specific signature of each of the regions. And more than 270 populations are now in our baseline, so quite a tremendous resource. There were some technical issues and standardization things that I'd like to talk about that were kind of interesting. We all use this technique called the polymerase chain reaction, which won a California surfer a Nobel Prize. He's the one that invented this technique. And actually my professor, Dennis Hedgecock, was one of the early professors to learn this technique. He went to sabbatical in the company where this technique was developed. Now, that's quite a sophisticated technique, and you can imagine that different laboratories from California up into Alaska may have some different means by which they achieve that complicated technique. We had to talk about all our different techniques and decide which ones we would use in common. And then we discovered also that we each had different microsatellites that we preferred to use. So we had to decide amongst more than 120 different microsatellites which were the best 13. And of course we all had different opinions about that. You might ask, so what is a good microsatellite? Obviously there are different parameters of interest here. One is how easy is it to amplify and score. Obviously that's something that needs to happen. The second major thing is how many different characters does it most commonly reveal among the different populations? And are those characters different in different populations? Although there may be many different characters, if they're not very variable among the populations, then it's not much use. So unfortunately, these parameters are kind of quite often in opposition with each other. Markers that are very easy to amplify might not have many different characters. Markers that have many characters and are highly informative across populations might be very difficult to amplify. And as I was just describing, because I was involved in the early isolation of these microsatellites, I was one of the investigators that I guess had experience with a lot of them. And I did pretty well in that. Actually 7 of the 13 in the baseline are out of the lab I used to be in and out of what we're developing here. And it was kind of interesting for me because amongst those 7, there were 2 that were not my favorites. Although they came out of my lab, they weren't nearly as informative. I knew that these markers were good and they were easy to amplify, but I knew that there were others that had been developed more recently from other labs, actually, that were more informative. And I strongly said, you know, although I'm shooting myself in the foot, you know, we need to get rid of those 2 Banks markers and replace them with something else. But because many of the other labs really liked them and they were easy to amplify, I wasn't able to succeed in shooting myself in the foot. So that's how it is now today. We all, I guess, see the world from our own particular windows. So here I am in Oregon, and I have also experience in California, so I'm very interested in markers that do well in Oregon and California. Someone in Alaska is more familiar with how markers are in Alaska. I guess that's why we have different opinions, and I think someone said, well, Michael, even although there are only 2 alleles in OTS3 in California, So it doesn't tell you anything about California fish. There are 15 up here in Canada or Alaska. And so we like that. I would guess that it probably took us about a year and a half to 2 to actually go through that decision process. Once we had decided on those 13 best microsatellites, we also had to ensure that we were scoring those 13 microsatellites correctly in California, Oregon, Washington, Canada, and Alaska. And we all have our own really high fidelity machines to score them. And we found that although these machines may cost $300,000 or $400,000, they don't necessarily always give you the same call for the same marker. So we had to do a process we call calibration, where we make sure that a particular genotype scored in California is scored in the same way as the genotype scored in Alaska and Canada and Washington, Oregon. What we utilized to check ourselves on that was what we call a blind test, where we had a tray of 95 fish that somebody else prepared and then provided to us, and we all scored them in each of our independent labs, and then we collectively submitted our results to a different party who saw whether we scored all the same.
Once the calibration process was complete, the average genotyping accuracy by laboratory and microsatellite was 99.4%.
We now have determined unique genetic signatures of the salmon populations up and down the coast. We've established a broad spectrum standardized baseline across the Pacific Northwest, which is just fantastic for its enabling power. We can now have fishermen go and catch fish anywhere in our Pacific Northwest, have them deliver those to us as soon as they can, and within 24 hours we can tell them where we think those fish came from, which is pretty fantastic.